mechanism of how it happens, and this protein class called chaperones that help facilitate protein folding. And I talked a little bit about why this is so important, right? We want proteins to fold properly so that they can have the right function, right? But it's also because if proteins fold improperly, they can aggregate. Because a lot of times, right, we talked about how the hydrophobic moieties, if they're exposed on the outer surface, what happens is they freak out because they're the cytosol is aqueous. So what happens is they can form these big aggregates with any other hydrophobic moieties. It's basically kind of like an intracellular tumor, right? These proteins just aggregate and they can impair other processes in the cell. And so protein aggregation is the cause of a bunch of different diseases. A bunch of, like, I know a lot of the neurodegenerative diseases is involved in, like, Alzheimer's, right? That there's a lot of protein aggregation that occurs in Alzheimer's patients. There are a lot of metabolic diseases that are the result of protein aggregation. So it can totally impair the ability of cells to function when you get these big protein aggregates. So it's kind of, right, twofold, to make sure proteins can form their appropriate structures and have their normal functions, and then two, prevent protein aggregates from forming and mixing up all the functions in that cell. So we talked about last time the two of the cytosolic chaperones, HSP70, which binds the hydrophobic moieties during protein synthesis to help give that protein a chance to fold properly. And then HSP60 comes in a little bit later, because this is important after a peptide has been fully translated, right? So the peptide ribosome is done translating it, and this is kind of a second line of defense. HSP60, what was the, is 60 cylinder, was that the 60 cylinder? Jazz hands, 60 cylinder. It forms this isolation chamber to give the peptide a chance to fold without other proteins or molecules getting in its way, right? So it can fold kind of on its own. And protein folding, it's not like it just folds and it's like, we're done, we're good here. It's kind of, it's an iterative process. So there's all these molecular interactions happening as it forms its alpha helices and its beta pleated sheets and all that rich, tertiary structures. And so the protein's trying to get it right. It's trying to minimize its free energy. And so the chaperones try to help it do that properly. And so we have a lot of proteins get translated on 3 ribosomes, right? And those would be the ones that are relying on HSP60, HSP70, because they're folding in the cytosol, right? But some proteins, I mentioned, if they're translated on a bound ribosome, and when they're translated, they're generally moving across the ER membrane, either into the ER lumen or into the ER membrane itself. So there's proteins that are going to be folding within the ER, right? So those proteins that are folding within the ER need chaperones as well. So the 2 of the really important chaperones in the ER are called calnexin and calreticulin. And so these proteins, the way they work is that they bind the proteins as they're folding in the ER lumen, and they essentially keep them in the ER lumen until they're properly folded. So, so the proteins that fold in the ER lumen, they're always going to be moving out of the ER lumen to the Golgi to get processed. Did you guys learn about that processing mechanism in Bio 2 at any point? So, okay, you move, and we're going to learn about the details of it in subsequent lectures and in detail in lecture 12-13. So they move from the ER lumen to the Golgi for further processing, and then they get directed out to where they're going to live permanently, right? And so all these proteins that are folding in the ER lumen, they're going to leave it. And so what the calnexin and calreticulin do is they retain them in the ER lumen to make sure they fold correctly before they're released from the ER. Okay. And there's a really interesting mechanism. So I'm just giving you the intro to, hey, these are these chaperones, and their mechanism is to hold the proteins in the ER lumen to give them a chance to fold properly. So they're just retaining them there. And they have this mechanism that involves the glycosylation. So this blue thing so this green thing is supposed to be a peptide, and this blue thing is the oligosaccharide that's attached to the peptide. So later in this lecture, we're actually going to talk about glycosylation that occurs in ER. So they get these big sugars added to them. And basically, the way that the protein folds, I wonder if I should go, I'm not even testing you guys on this, so I don't know if I should go into this level of detail. But essentially, there's a mechanism in which the cell or the proteins can detect, according to the location of the sugar group, if the protein is folded properly. So the calnexin and calreticulin are kind of involved in that sensing mechanism. If you want to learn more on the slide, I actually kind of pasted a paragraph that talks about how that mechanism functions. For the purpose of the exam, really what's written here is what I want you to know, that level. There's always so important. There's always so much more to learn. It's limited. 1 hour and 15 minutes, which really is already longer than most of our attention spans. Okay. So what happens if a protein doesn't get it right? What happens if, even with all the help of the chaperones, right, the protein still doesn't fold properly? What happens is it gets released into the cytosol, and it gets polyubiquitinated. So I mentioned ubiquitin when I was talking about different ways. I think I introduced it when I was talking about different ways for histones to be covalently modified. Ubiquitin is a really tiny protein that can be added to really any protein. It's a type of covalent protein modification. But when it's involved in protein degradation, not just a single ubiquitin gets added, many ubiquitins get added. So that's called polyubiquitination. So, right, that's why it looks like a boxing glove. That's why there's many boxing gloves. There's many ubiquitins being added to this peptide. That is the signal to the cell that that peptide needs to be delivered to the proteasome. The proteasome is basically like the garbage disposal of the cell. It chews up proteins and spits them out. So this is the way that a cell, right, invests all this energy into trying to get the protein to fold properly, but it also knows if it doesn't get it right, it needs to degrade that sucker before it potentially causes an aggregate, right, and destroys the cell's function. So for any protein that is misfolded, this is supposed to be the fate, right? It is supposed to get polyubiquitinated and degraded by the proteasome. Right, in diseases, when protein aggregation is the cause, this isn't happening normally, right? Again, everything I talked to you about the way that the cells function, there is some disorder that is caused when that function breaks down. This is all relevant to health. It's interesting, right? Or it's not interesting, but it's frustrating because I think the most captivating thing is to show some image of some crazy disease and be like, this is what breaks down when this disease happens, but you can't understand that until you learn how things work normally, right? Okay, so normally proteins are supposed to be broken down by the proteasome if they fold them properly. And the reason for that is because they can aggregate and totally destroy the function of the cell. So again, protein folding is quality control, right? And so the chaperones are basically working against the clock. So there's a lot of words, so I'm going to read that so you can listen to what I'm saying instead of reading. So no quality control needed for correctly folded proteins. So some proteins, they get it right on their own, right? That's what they're showing over here. It's correctly folded. If it correctly folds on its own, cool. Chaperones don't need to expend their energy. Incompletely folded proteins are refolded by molecular chaperones. So if it gets off track, and usually that means it starts exposing hydrophobic residues, chaperones are like, let me help you try to get back on track. So the protein rescue process that chaperones participate in are competing with destruction by the proteasome, right? They're trying to get it right, but if the misfolding is too bad, or if it can't get it right after a certain amount of time, there are basically degradation signals. There are proteins that will bind and mark that improperly folded peptide for polyubiquitination. It gets polyubiquitinated by addition of all those boxing glove ubiquitins and then sent to the proteasome and destroyed. Right? Processes prevent protein aggregation. It occurs when misfolded proteins clump together non-specifically. So again, right, it's that relationship analogy. You have a long-term relationship, you invest so much transcription, translation, and now we're folding. The chaperone is like the counselor to try to help you out. Let's try to make it work. But if it doesn't work, right, this is a good lesson for all of you. At some point, you just gotta trash time. I learned that lesson the hard way in my 20s. Oh my gosh, mistakes that I made, and that is a super TMI conversation, so I'm not gonna have it. But I still remember when I met Jack, my husband. It was like within a month or two of dating, I was like, I don't understand. There's like nothing to fix. I don't. Oh, this is what a functional relationship is. This is amazing. I've had one before. There's a lot of weird people in the world. Okay. So this is, again, just reiterating this idea of, right, a newly synthesized protein increasing time, right? So the quickest and most ideal situation is protein can fold without help. Awesome. It doesn't always work that way, right? Like nothing is 100% efficient. So it takes a little longer. We need the help of a chaperone, okay? If that doesn't work out, right, that protein is on the clock. And so ideally, that incompletely folded protein that the chaperones, there's nothing they can do, they get digested by the proteasome. And when that fails, in the few times that it fails, it can cause protein aggregation, right? Very bad. It can cause debilitating disease. There's all sorts of diseases that are caused by protein aggregation. Okay. And so the proteasome. Right? I mentioned that it's regarded as like the garbage disposal of the cell. So let's look at the proteasome, unless you guys have any questions on this. Yeah?
So is the proteasome complex and the chaperones, are they competing against each other? Are they both competing to grab the improperly folded peptide?
Yeah, essentially there's a mechanism by which the it's kind of set up so that the chaperones, in a sense, get first dibs, or you have a scenario where if it's just too misfolded, right, it's not going to be able to outcompete like the degradation signals that will bind and target for polyubiquitination. So it's kind of there's a lot going on with the trying to get it folded properly, but making sure that a misfolded protein doesn't get to persist. So yeah, you can kind of think of it as chaperones and then the degradation signals as essentially being in competition with one another, but it's all really to maintain the viability of that cell. Like, try to get it folded properly, but if there's just not working out, gotta make sure we get rid of that protein. Okay, so misfolded proteins are ubiquitinated, and not just ubiquitinated, right? Polyubiquitinated. There's a bunch of little ubiquitin molecules that get added in succession, and that targets them for destruction by the proteasome. So the proteasome is a big old protein complex that at its what's regarded as the central cylinder. It has a bunch of different proteases with the active sites all facing the core, right? So basically the part of the protease that binds the proteins and cleaves it, they're all facing the area where the protein's going to move through. So basically when a misfolded protein moves through, it's not just cleaved by one peptide, or it's not just cleaved by one protease, there's a bunch of proteases ready to chop that protein up so that by the time that protein exits the other side, it is just in tiny little pieces. And so essentially the amino acids can be recycled by itself and reused for other translation events. Okay. Questions? Good. Okay. So now we're going to talk about membrane insertion. So there are a lot of proteins that end up embedded in membranes, right? We have proteins that are globular, and they hang out in like the cytosol or the lumen, or they get secreted. There are a lot of proteins that live and function within membranes, right? So transmembrane proteins. How do they get inserted into membranes? Okay. So the way they get inserted, first, it is always through this protein that's called a protein translocator. Is that on this slide? Maybe I get to it on the next slide. But it is one more. Okay. That is the protein translocator in blue, right? This is the membrane protein itself. That helps facilitate movement of peptides through a membrane or to embed them into a membrane, right? Here it's membrane insertion, we're inserting it into the membrane. We're going to talk quite a bit about protein translocator. All right, so what the heck's going on in this image? Green, that's the ribosome. Blue, what's blue? It's the RNA, it's the mRNA. And then right here, this phallic-looking thing is the peptide. So the ribosome is translating the peptide. And what this image is trying to show is that some peptides are translocated across the membrane as they're being synthesized, right? So that's referred to as co-translational membrane insertion or co-translational translocation. So co-with, right? During translation, it's translocated across the membrane. That's what happens most of the time, but there are some membrane proteins that get fully translated prior to being inserted, right? So after translation, so post-translational translocation. So we know, so both, oh there it is, both methods use a protein translocator, which is again, just stay in blue. There. And so in eukaryotic cells, most membrane proteins are co-translationally translocated unless you're a membrane protein destined for the mitochondrial membrane, peroxisomal membrane, so peroxisome is an organelle, or the nuclear membrane. So transmembrane proteins that go to those three targets, mitochondrial membrane, peroxisomal membrane, or the nuclear membrane, they are fully translated first and then inserted into those membranes. Okay, so far? Alright, so how does the protein translocator work? It recognizes ER signal sequences. So it's called, they're signal sequences. Oh. Just found a typo on my own slide.
Yeah, she said T-M is transmembrane.
Sorry, I'm so neurotic about this. I'm freaking out internally right now with this typo. Okay, if it is going to the ER, it is going to have an ER signal sequence. If it is going bound to the nucleus or the peroxisome or the mitochondria, it has signal sequences that target it for those membranes. Sorry, I'm internally giving myself an F. Okay, and it's okay, it's gonna be okay, we're gonna do okay, so, but the sequences are always referred to as start-stop transfer sequences. So it indicates to the protein translocators that this is where you need to start transferring the sequence across the membrane, right? And so there's this really cool mechanism for when a ribosome binds to the ER and initiates translocation. The ribosome basically kind of passes off the peptide to the protein translocator. We're going to learn in detail about that mechanism in lecture 12-13. And so the protein translocator itself recognizes these sequences that indicate here's where you start moving this peptide across the membrane. And those sequences are called start-stop transfer sequences. They tend to be, they're spanning the membrane. So what would you guess? Do you think those are hydrophobic or hydrophilic stretches? Hydrophobic, yeah, they tend to be really hydrophobic stretches, right? Because the membranes, the lipid bilayer, they're lipids, right? And so these start-stop transfer sequences tend to be very rich in hydrophobic amino acids. And so it can go either way in terms of the direction it inserts the peptide across the membrane. So you can end up with the amino terminus in the ER lumen or in the cytosol. So there are various examples that go in either direction. And this is an example of an internal ER signal sequence. A lot of times they're actually at the very N-terminus of the protein, and so that's what's shown on the answer was that's what's shown on this slide, where you have this N-terminal ER signal sequence that is the first thing that gets recognized by the protein translocator, and it weaves the peptide across. So that's like the start transfer, right? It's starting to transfers until it gets to this subsequent stop transfer. So it stops transferring, translation still occurs. So and we'll go over this mechanism. A lot of times that first N-terminal sequence is cleaved off. So you end up still with this single pass transmembrane protein. Right? But the protein translocator, it basically needs to be told what to do by the peptide. So the information, that's kind of the take home, is the information is in the peptide. The peptide has start transferring now, protein translocator, okay. So it binds and the translation still proceeds, and then if it gets to a stop transfer, it goes, oh, okay, we'll just embed this part in the membrane too and continue translation, and if there's another start transfer, that will start getting transferred across the membrane as well. So, right, because a lot of times you have, yeah, go ahead.
Does that mean that so you're saying that the start sequence gets taped off at the end?
Yeah, sometimes it does. So in this example, when you have an internal sequence, it doesn't. But in many of the cases, and in fact, in the beginning, when we first realized that how this mechanism worked, the first identified ER signal sequence was identified at the N-terminus. And so for a long time, we thought, oh, they're only at the N-terminus, and they always proceed by this mechanism where it gets cleaved. And then we started identifying proteins that proved that wrong, right? That wasn't the only way. This happens all the time in science. It's a beautiful thing, right? We shouldn't think we know it all. We should never think we know it all, because we never ever do. We know a lot, but there's always new information. And so all of a sudden, these proteins were identified that didn't have that special N-terminal sequence. It was actually embedded kind of right smack dab in the middle of the peptide, and that was also recognized by the protein translocator, and it moved it through no problem. Okay. there's always exceptions to everything. I could start every one of these lectures saying there is an exception to every single thing I'm about to teach you. There are exceptions all over the place in biology. And I wish I had time to teach the exceptions and the rules, but I don't. I get tired, more coffee, work out. But
So this one's showing co-translational regulation, right?
yeah.
For post-translational, does it still go through the same process?
Yes, so post-translational, that is a great question. Yes, it still requires a protein translocator to help move it across. And we're going to talk about, I don't know what lecture, but in one of the lectures, we're actually going to look at the translocators that exist within the mitochondria to help facilitate that. And so they're called TOMs and TIMs, translocase of the outer membrane, translocase of the inner membrane, and so, or translocator of the outer membrane.
So there's like still stop and start sites in
Yeah.
the post-translational?
Yeah, so anytime you're, thank you, because too many words are coming out of my mouth. So to trim it down, right, the take home is if a protein wants to get embedded in a membrane, it will have at least the start transfer, and if you're, and oftentimes also a stop transfer sequence, right? So it is the way that the peptide associates with these protein translocators, which is the general name of these proteins that sit in membranes and help embed membrane proteins in the membrane. Okay, right? So if you took this protein right here and you somehow disrupted it so it didn't have the start transfer or stop transfer sequence, it is not going to get passed through that ER membrane. It's going to end up living in the cytosol. I mean, it'll probably end up getting degraded. It's probably going to fold all funky. Right, but that's the idea. These sequences are what associate with the protein translocator and instruct it to move it through. And that's what, a lot of the crux of a lot of the next few lectures is all about peptides having their kind of targeting information embedded into their sequence. They tell the cell, once they're translated, I need to go to the mitochondria. I need to go to the ER. I need to be secreted. They have these sequences and structures embedded in the sequence that tell the cell where to take them. They're housed with their own address, right? So we're going to talk about these different addresses and mechanisms that the cell uses to get the proteins where they need to go. Right, that's the next thing in this process. We talked transcription, translation, talked about protein folding. So now we need to get these proteins to their home, right? They want to go where they need to do, or they need to work, whatever. They need to go someplace and do their job, and we need to figure out the mechanism, or I need to teach you the mechanism by which a cell gets these proteins to the proper destination, right? And here's if they need to get into the membrane. Here's step one, right? Yeah.
They have another
Summer? Yes.
Did they have another sequence, like a start sequence? Would that one get cleaved as well? Or would it?
Typically, it's only like the N-terminal one that will get cleaved. Yeah. And so, right, the next slide, there are a lot of 7 pass transmembrane proteins. So 1, 2, 3, 4, 5, 6, 7. This has to weave back and forth, right? So this has start transfer, stop transfer, start transfer, stop transfer, start, stop, start. Does that make sense? Okay. All right, so another thing that's really important for protein function is something called covalent modification. This is introduced when I talked about histones, right? What kind of covalent modifications did we talk about with histones? Methylation. Yeah, methylation. So the take home with covalent modifications are always performed by an enzyme. So an enzyme has to add the covalent modification or help participate in the covalent modification. And oftentimes they're reversible, sometimes they're not. And the covalent modifications that I'm going to introduce right now, there are a ton of them. I'm just going to introduce a few of them. Glycosylation, so there are glycosylating enzymes that add these big old sugar groups to proteins. Anytime you see these hexagons in the images in this class, those are always representative of glycosylation. Kinases. Kinases phosphorylate proteins. Does anyone know the name of the protein that removes phosphate groups? Phosphatase, yeah. So there's cells are full of different kinases and different phosphatases. And they typically have a lot of really specific target proteins that they will phosphorylate or dephosphorylate. And so, again, right, we're talking about a covalent modification of a protein, which means that the sugar group or the phosphate group is being added to the side chain of an amino acid, right, by the addition of a covalent bond. So they're really stable additions to these proteins. And if you're adding a phosphate group, you're adding a sugar group, at the end of the day, why does this matter? What is this changing about the protein? What can it influence? Function, right? Because you're changing structure. You're adding a whole other something to the protein, right? That can influence structure, change function. Another big one is disulfide linkages by types of enzymes called protein disulfide isomerases. Does anyone know the name of the amino acid that has a sulfur group in it? Cysteine, you got it, yeah. And so cysteines form these really strong disulfide bridges that can really influence the way proteins function. I think we talk about it in the context of insulin in this class. A lot of examples. In fact, the speaker, right, my advisor, he's famous for Wnt signaling. Wnt proteins, their structure is totally reliant upon these disulfide bridges. So in this protein family, they typically have like 17 to 19 highly conserved cysteine residues that all form disulfide bridges. You lose those, you lose the ability of Wnt to bind its receptor and influence stem cells and development, all the let's do everything. All right, and then some of the enzymes that we've already introduced, methylase and acetylase proteins, right, that can add methyl groups or acetyl groups to proteins, and you've learned about how the powerful effects that can have on gene expression when there's an insulin. But tons of proteins can get methylated and acetylated. And in lecture 10, which is a lecture I'm supposed to be on right now, I go into much more detail about how the cell kind of regulates the function of the proteins by manipulating the enzymes that perform these covalent modifications. Okay. Polymer assembly. So polymers are macromolecules composed of repeated subunits. One of the polymers that we're going to talk about a lot in this class, post-exam too, is microfilaments, which are basically actin polymers. And we're going to talk about microtubules, right? These are cytoskeletal proteins. So microtubules are composed of tubulin polymers of tubulin dimers, right? Actin on its own, meh. Actin, when it's polymerized in the context of a microfilament, has dramatic implications for the structure of a cell and the way it functions. The example shown here is collagen. So an individual collagen protein forms this rope-like superhelix. And it turns out, right, it's important for the extracellular matrix. Triple helix. They wrap around each other to form this braided conformation. But when you find collagen in the extracellular matrix, the collagen that matters and it's functional, so here's some images of the braided collagen. When you see it in the extracellular matrix, these braids basically polymerize with one another. So they form linkages. So you get these big old collagen polymers where it's braid attached to another braid attached to another braid. And that's what really gives it its tremendous kind of tensile strength, right? All these braids that are rope-like and super strong, and they're tethered to other braids. So a polymer, right? Poly, multiple or many, and mer, units. So if you hear polymer, it just means there's many subunits of whatever you're talking about. Okay. So a lot of proteins really only function when they're in the context of a polymer. So we're going to talk about how cells manipulate polymer assembly to manipulate the function of those proteins. And proteolysis, right? Oh, here's the example of insulin, and so you can see the sweet disulfide bridges that are forming. So insulin, when it's first produced, it's formed as a proinsulin. So this is it's not functional yet, and so it has to perform this really specific tertiary folding, right, which involves the formation of these disulfide bridges, and then it gets proteolytically cleaved, and only then do you have your functional insulin molecule. So proteolysis is critical for the function of insulin. You need it to happen. So one thing about proteolysis is very novel with relation to some of the, or all the other covalent modifications I talked about. It's irreversible. Once insulin is cleaved, there's no mechanism in the cell that says, oh, we need to find that gray piece and tape it back on just so. That doesn't happen. Proteolysis is irreversible. So if there's the question on the test, like which of the following is not reversible? Proteolysis. That's the answer, right? Proteolysis is one of the only irreversible modifications. One of the other irreversible modifications that we talked about, does anyone remember it with respect to DNA? Methylation. Methylation, yeah. Okay. Yeah.
So is this considered a covalent modification?
Yeah, because you're affecting the covalent linkage between the peptide bonds. Yeah, so that's why it's categorized as a covalent modification, and it's typically performed by specific enzymes that cleave those specific bonds. Does that make sense? Yeah. I know, when I first saw this, I'm like, is that? And yeah, that's how the textbook rationalized it. For other professors who designed this curriculum originally. I'm racking my brain. What is your name again?
Christina.
Christina. Orozco? After I pass back the first exams, I try so hard to remember, but 120 is a lot. Try. Amanda? Sorry, okay. Oh, okay, diabetes. Yeah, there's two different types of diabetes, right? So there, type 1, there is a genetic problem. They don't make their beta cells, which is the cell type in the pancreas that makes insulin, are either, I think it's an autoimmune thing. I think their own body attacks the beta cells, and so they don't produce enough insulin. And then type 2, right, which used to be called adult onset, but so many children are afflicted by it now, that is, what are they calling type 2? I think it's just type 2 diabetes as adult onset. That's different because it's acquired, and I'm forgetting the mechanism by which that occurs. It seems like there are genetic underpinnings of that as well. So I've heard a really interesting program, I think it was like on Science Friday on NPR or something, but they were talking about how there's this social stigma against people who have type 2 diabetes, like, well, you did this to yourself by not being active enough or not or eating McDonald's every day, all day, or whatever, but whatever people think. But it turns out that it seems like there is a genetic predisposition for type 2 diabetes as well, but we're still trying to figure out what that is. Okay. Holy crap, I am gonna get to lecture 10. So I have an activity that I want you guys to work through on your own at home and let me know if you have an issue. This was kind of part of the activity I put on the learning module, and it gets back to alternative splicing. I just really want to make sure that you understand how that proceeds, and I think sometimes by drawing it out and trying to get you to draw it out, it helps. But I think I got at this in the quiz, right? If you have two cells where they splice the same, they both express the same pre-mRNA, it's exactly the same, right? And in this case, they have 3 exons and 2 introns. What is it that's different in those two cells? Is it the splicing enhancer, the spliceosome, or the splicing recognition factors? It's the splicing recognition factors. They're going to have the same splicing enhancers because the pre-mRNA sequence is identical, right? And the splicing enhancers are sequence on that mRNA. It's about are the recognition factors present to bind those sequences and then recruit the spliceosomes there. Spliceosomes are the same too. Do that.